Respected resource person of the day, Dr. Elizabeth Cherian, Dr. R. Nishan Nair, Head of the Department of Botany, St. Joseph's College for Women, teachers, delegates from different parts of the country, and my dear students. Recerca 2020 is intended to provide a common platform for students to share research ideas and research updates and involved in discussion from critical perspectives and improve presentation skills. We expect that this event will motivate the participants to undertake interdisciplinary, multidisciplinary, and transdisciplinary research in future. Today, as we reach the fourth day of national level project presentation competition, Recerca 2020, it's my pleasure to welcome you all to this session. First of all, I would like to introduce and welcome today's subject expert, Dr. Elizabeth Cherian, Assistant Professor, Department of Botany, CMS College, Kottayam. She has 15 years of research experience and serves as research guide in the Department of Botany, CMS College. She has completed many research projects and published many papers and books. I warmly welcome Dr. Elizabeth Cherian, who wholeheartedly accepted our invitation to judge the presentation today. Welcome, ma'am.
Thank you, Diana.
We have 14 selected participants from various colleges and institutions from Kerala and also from outside Kerala. You are the essence of this whole program. I am happy to extend a very warm welcome to you all. Let me welcome Dr. R. Nishan Nair, Head of the Department of Botany, who is always an inspiration and support to us. Welcome, ma'am.
Thank you.
I welcome the teachers and students of St. Joseph's College to this program. Once again, welcoming you all to this program. Thank you. So before we go further, I would like to repeat some instructions. Before we go further, I would like to repeat some instructions already given to the participants. Only 8 minutes will be allowed to present and 2 minutes for discussion. In the case of group projects, during discussion, group members can also answer. Then while presenting, other delegates must mute the audio and video and pin the presenter slides. Then recording is not allowed without the permission of committee. Then abstract will be published as proceedings and soft copy will be provided when ready. The decision of subject expert in result will be final. For getting a certificate, feedback form must be submitted correctly without mistakes. Then the Excelsior Award will be announced in the validatory session only when presentation of all subjects are completed. Now I request your active participation throughout the session. Thank you. Moving on to the presentations.
Good afternoon to all. Myself, Smridana Ramesh, and I am presenting my project entitled Mutagenic Action on Seed Germination and Growth in Abelmoschus esculentus. As human populations increased, greater and safer supplies of food had to be found. And we all know that mutation breeding is a strategy for the enhancement in food production, in which mutagenesis is used to produce genetic variation. The present studies have provided and the effects of various mutagens on Abelmoschus esculentus, which is commonly known as lady's finger. Lady's finger, we all know that an economically very important vegetable crop. It is an excellent source of vitamins C and K1. It contains beneficial antioxidants. It is linked to blood sugar and cholesterol control. It contains a protein called lutein, and it is being studied for its role in cancer prevention and treatment. It may help pregnant women to meet their daily foliage needs. And clearly, I'm aimed to find the effects of various mutagens on the crop mentioned. Moving to the materials and methods, the variety used here is Varsha Upahar, and its good quality seeds were collected from Rice Research Station, Mangombe. And the mutagens used here includes UV radiation, which is a physical mutagen, sodium azide, ethyl methane sulfonate or EMS, colchicine. These are chemical mutagens. UV radiation is applied in three different durations, which is 15, 30, and 60 minutes. Then chemical mutagens are used in three different concentrations. They are zero point 0.1, zero point 0.4, and zero point 0.7 percentages. Hence, the each treatment, the plants of each treatment is represented as the mutagen applied on it and the percentage or duration, like UV15, UV15, EMS1, SA4, COL7, like that for convenience. First, primarily the seeds were surface sterilized by 0.01 percentage mercury chloride for 5 minutes, and it is thoroughly washed with distilled water for several times. It is then pre-soaked in double distilled water for 10 hours to initiate metabolic activities. Treatment is done only after pre-soaking. Then, after pre-soaking, the pre-soaked seeds were blotted dry, and for UV treatment, the seeds were placed under UV light for the different aforesaid three durations. And the chemical treatment is done in freshly prepared phosphate buffer of pH 7, and it is for 6 hours. Then, after the treatment time, the seeds were thoroughly washed in running tap water and it is blotted dry. Then the seeds were sown in trays in an experimental field. Then the data were recorded in various parameters like seed germination, plant height, number of leaves, and so on during the successive days. These are the major steps within a single slide. Okay. Then I got the following results. Regarding seed germination and seedling growth, the control or untreated plants and the UV treated plants were first germinated. Other treated plants had a dose dependent decrease in germination. UV treated plants exhibited a mixed effect in terms of seed germination. SA7 and colchicine treated plants had not germinated. Then I repeated the colchicine treatment by reducing its concentration to 0.01, 0.04, and 0.07 percentages. Then it had a 30 to 40 percentage of germination. In plant morphology, on 10th day, UV30 and COL1 plants had more height over control. And also, control and UV30 plants had larger and 4 average leaves. Other plants had less 4 leaves. On 20th day, control shown more height than others. Control and UV60 plants had 6 average leaves. Other plants had less than 6 leaves. When the plants had 20 to 25 days of growth, it had been attacked by mealybugs, but it was less on EMS treated plants. That means the EMS treated plants developed a sort of resistance
Hello?
control. UV30 plants were good in growth. More average leaves were found in UV60 and EMS1 over control. On 40th day, UV30 were larger than the control. Some of the UV60 plants exhibited vigorous growth and more leaves over normal. In viewing survival till maturity or the flowering stage among treated plants more survival exhibited by UV30 plants. It exhibited 80% of survival. SA7 and colchicine treated plants were no more. They were not survived. Then, in viewing this table, you can clearly understand that about the flowering period. UV30 plants were early flowered. They started flowering 35th day onwards. Then only the control plants flowered. And SA4 plants were the last flowered ones. In flower morphology, flowers on UV30 and UV60 plants had larger than control. Flowers on UV60 and EMS1 and also SA1 had variation in the number of petals and stigmas. Because also, these are the flowers on UV30, UV60 and control plants. See the difference in size, flower size. Okay. Then according with the yield, the UV treated plants were good in yield. Here I am two figures presented. In both of them, C, D, and E represents the fruits from UV treated plants. See, UV treated plants produced more fruits. I conclude that UV treated plants had highest germination and survival with other treatments, and EMS plants were resistant to mealybugs. UV60 in some of them exhibited vigorous growth. UV30 plants had early flowering, more flowers, and more fruits also. These are some important references that help me so much, and I express my respect to everyone who helped me to do this work and to present like this as well. Thank you. Thanks to all.
I appreciate for your nice work.
Thank you, ma'am.
Let me ask some questions also. Actually, by selecting this work, what was your actual or main aim to increase yield or to increase the nutritional value or to pest control, or what was the main thing?
Okay. Which is what is the actual aim? Is that you told? Okay.
To which factor you gave much priority? That's what I mean.
Okay. I think mutation breeding program helps to produce new varieties, new plant varieties with more desirable traits. Okay. Through mutation breeding. In my work, I'm explained. I'm trying to find the effects of various mutagens on this particular crop.
Okay.
And if one mutagen is found suitable to that crop, that mutagen can be used to produce a good variety of that crop.
Okay.
And anyone doing this mutation breeding program who can use that mutation, that particular
by
mutation.
saying variety, excuse me, by saying variety, what do you mean?
A new variety,
was,
a new genotype.
Okay. Favorable in which sense?
Favorable in particularly the yield.
Okay. It's a food source, so we have to give some importance to the nutritional value also. That's why I asked.
Okay.
You have mentioned its nutritional value for its.
Actually, I am observed so many factors, so many parameters I'm observed, and actually I'm already planned to do the cytology works also if there is a change in the crop.
Okay, nice.
But due to the COVID-19 pandemic, I can't do the work as complete. Yes, I've already collected the pollen grains. Then, if
you
condition
get some more
permits,
time, you can do that.
Okay, if the condition permits, I will definitely continue that work.
Okay, good. Thank you.
Okay, ma'am.
Okay, now we'll move on to the next candidate. I welcome Aksara IG to present her work. All the participants are requested to switch on the video while presenting.
A warm good
Okay.
afternoon to one and all present in this virtual platform. I'm Apsara IG and I am here to present my PG project on production of enzymes by endophytic bacteria of medicinal plants. So from the name itself, we can understand that endophytes are those organisms which are present in internal tissues of the host plant without causing any harm to it. So it basically in plants it constitutes the fungi and bacteria. So you may be thinking why these organisms are very important. Mainly because of the benefits that it render to the host plant, like nutrient supply, then production of secondary metabolites, plant growth promoting substances, then host defense against biotic and abiotic stresses. It can also be used as biofertilizers, biocontrol agents, etc. But I have focused in this work on the enzyme production by these endophytes, and I have concentrated on three important enzymes, that is proteases, which cleave the peptide bonds of protein, then silanases, which catalyzes the hydrolysis of beta linkage of cellulose, and cellulases, which cause the degradation of cellulose. Coming to the aims and objective of my project, first of all, isolation of two bacterial endophytes from the rhizome of Maranda albinaceae, a monocot plant, isolation of two bacterial endophytes from the roots of biophyton syncytium, a dicot plant, morphological as well as molecular characterization of the isolated strains, identification of the strains using bioinformatics tool, then localization of the endophytic bacteria using a novel tissue clearing procedure, then screening of isolated endophytes for enzyme production. Coming to the materials and method, considering the nutritional as well as the medicinal importance, I have chosen rhizome of Maranda ardidaceae and roots of Biophyton syncytium for my study. And first of all, for the isolation process, I have surface sterilize a plant material in a combination of 70% ethanol and sodium hypochlorite. And after that, the control plates and inoculate plates were incubated for about 48 hours in 30 degrees Celsius. At the media composition that I have used is given below, like agar 2.2 gram, NaCl 0.5 gram, peptone 0.5 gram, and yeast extract 0.5 gram in 100 ml distilled water. So the isolated bacteria were analyzed for morphological characters such as color, elevation, opacity, gram staining nature and all. And for molecular characterization, first of all, genomic DNA was isolated and PCR analysis was done using genus rRNA primers, and finally the sequence were obtained. And the obtained sequence were compared with already present sequence in the GenBank database, and the multiple sequence alignment was done using cluster next to offline and muscle online, and phylogenetic trees were created and viewed using NJ-plot. For the localization of bacterial endophytes within the tissue, first of all, a novel strategy was used, that is, the tissues were first of all cleared and then gram staining was performed. So the procedure that I have followed is given here. First of all, sample fixation was done in 4% paraformaldehyde for about 24 hours, and it was rinsed in 40 millimolar pipes. And after that, the plant material was incubated in a clearing solution containing urea, glycerol, Vitenex, and pipes. Then, after 4 weeks, I observed partially cleared tissue, and that was taken for anatomical sections, and gram staining was performed and observed under microscope. So for enzyme activity, I have analyzed the enzyme activity of three of silanese, CMCs, and protease activity of the endophytic bacteria isolated. For silanese assay, the substrate silane was homogenized in phosphate buffer, and after that, to this reaction mixture, enzyme preparation was added and incubated, and to terminate the reaction, DNS reagent was added, and absorbance was measured at 540 nanometer. For CMCs assay, the same similar procedure was used here also, but the substrate I used was CMC. And for protease assay, the substrate used was casein, and it was homogenized in phosphate buffer. And to this reaction mixture, enzyme was added and incubated for about 10 minutes at 40 degrees Celsius, and the reaction was terminated by adding 10% trichloroacetic acid, and absorbance was again measured at 280 nanometer. Coming to the results and discussion, and from Maranda albinaceae, I could isolate three bacterial endophytes, and it was named MAR1, MAR2, and MAR3. But due to the similar morphology of MAR1 and MAR2, I have discarded the MAR2. From Biophyton syncytium, I could isolate two bacterial endophytes that were named as Bio1 and Bio2, and all were studied for the morphological characters such as colony shape, margin elevation, gram staining, cell shape, etc. And for all the isolates were convex colonies with gram-positive rod-type bacteria. So this is the electrophoresis result of PCR amplification of genus RNA of the isolated strain. So after analyzing the phylogenetic tree, I could identify that Bio1 belongs to the Cellulose microbiome species, then Bio2, Cellulose microbiome aquatic strain Bio2, And MAR1 was identified as cellulose microbial species again, and MAR3 was identified as Bacillus corean strain MAR3. Only species level identification was possible for Bio2 and MAR3, so the partial sequence were submitted to GenBank, and I'm waiting for the GenBank ID. The localization procedure that I have used in partially clear tissue was successful, that I could observe purple color colonies in the anatomical section of rhizome of Marantha. So it indicated the presence of predominantly by gram-positive bacteria, and in biophytum I could observe both gram-positive and gram-negative bacteria. So screening of isolated endophytes for enzyme activity, first of all, CMCS activity that was maximum for Bio1, that's about 0.49 units per ml at 24 hours, and Bio2 also showed the similar activity, that is 0.46 units per ml at 24 hours, which is followed by MAR1, MAR2, and the least activity was shown by MAR3. Silanase activity was again maximum for Bio1, that is about 0.98 units per ml at 24 hours, and Bio2 also showed a similar result, and which was followed by MAR1, MAR2, and here also the least activity was shown by MAR3. Coming to the protease activity, again it was maximum for Bio1 of about 0.41 units per ml at 24 hours, and followed by Bio2, MAR2, and MAR3, and here the least activity was shown by MAR1. So comparing all the bacteria for enzyme activity, I could observe that the maximum activity was shown by Bio1 for all the three enzymes like CMC, CMCA, silanase, and protease. Coming to the conclusion, this is the first report on the isolation, identification, characterization, and localization of endophytic bacteria from Marantha and Biophytum. And I could identify 4 bacterial endophytes, that's MAR1 as cellulose microbial species, MAR3 was identified as Bacillus coreensis in MAR3, and Bio1 as cellulose microbial species, and Bio2 was identified as cellulose microbial aquatius in Bio2, and the partial sequence were submitted to GenBank. The localization procedure that was used in partially clear tissue was successful, that I could observe the gram-positive and gram-negative bacteria in the anatomical section, and the cellulose microbial species in Bio1 shows the maximum activity for all the enzymes like protease, cellulase, and silanase study. The further study can be done to identify the species of Bio1 and MAR1, so more markers have to be analyzed. Then visualization of these endophytes can be done confirmed using like STEM, confocal, and fluorescence microscopy in future. And antimicrobial as well as antibiotic studies can be done. Then secondary metabolic production analysis can be done, and also quantification and quantitative analysis of growth promoting substance can also be done in future. These are some of my references. Thank you for your patience. Thank you.
Okay.
Thank you, Apsara. Now the session is open for the discussions, ma'am.
Okay.
Apsara, is this your PG work?
Yes, ma'am.
Okay, you have done a lot and to do a lot. Okay, good work.
Ma'am, I have also done the growth promoting study also, but I couldn't complete it. I have just done
Okay,
the
okay.
preliminary study. Yeah.
How long you take to complete it?
Ma'am, I started the work at January. Then,
Okay.
and in between Corona time also, I used to go to college.
Oh,
Identification and all those
good,
things
good, good.
were done at that time itself.
Okay, okay. Okay, Apsara, then why did you selected these 2 plants? What was the criteria for selecting? One is a small, very small herb, and another one, what was the common name of Maranta?
Ma'am, arrowroot.
Okay, okay. I'm asking, is there any criteria for selecting these 2 plants?
Ma'am, actually, my work was focused on endophytes basically.
Okay.
Then I focused on medicinal plants. Like I have heard that the endophytes are responsible for the production of some of the secondary metabolites which are produced by the plant. So I focused on the medicinal plant. Then I reviewed the literature. So there was not even a single work was done on Marantha and Biophytum. So like in the sense, isolation of endophytic work was not at all done in these 2 plants. So I selected these 2 plants, and moreover, I need to study the growth promoting activity also. So I thought like selecting a monopod plant and dipod plant, if I could isolate an endophyte and inoculate it in a dipod plant, what will be the variation? Like I thought of studying that, but I couldn't complete it. Actually, I thought of studying that. Yeah, that's why I
Okay.
selected
So
only for endocyte.
you randomly selected this.
Yeah, looking at the medicinal importance first of all, then
There are
the
lots of medicinal plants.
yes, ma'am, but no work had been done on these plants like Maranta and Biofilm.
Okay, okay,
No
okay.
endophytic bacteria were isolated till now.
Okay. Do
So
you
that's
think this bacteria can impart some of the medicinal activities due to these bacteria also?
um, ma'am, I need to do work on that. Actually, that medicinal
You
property
might
like
have read the literature.
yes, ma'am. Cellulose microbiome. This is actually a pure endophyte, and that is involved. I have read that this is involved in used as biofertilizer. That is, it can
Okay.
promote growth. Yeah. So, is there any secondary metabolite involved in that? I need to analyze this, ma'am.
Okay, okay. How did you do that enzyme activity?
Ma'am, for submerged fermentation study, like liquid culture was done, and after that, like after 144 hours, it was liquid culture was done. And
What was the medium used for that?
ma'am, the same medium, that nutrient medium that I have like peptone, yeast
I'm asking this
extract.
because it's an endophyte. It is
Yes,
growing on a plant.
no, no.
So,
After isolation, I have done this fermentation study. Like
okay,
isolation,
okay,
I have
okay.
already. Yeah, I have that
Yeah,
bacteria.
I'm asking what was the medium used to culture these microbes,
Yes,
the
ma'am.
isolated microbes.
Like for isolating, I have used the agar media, the semi-solid media containing agar, like yeast extract, peptone,
Plus,
then, huh?
yes,
Sorry, ma'am.
plus agar plus what all the other ingredients?
MgCl2 was used and NaCl was used for the like for the isolation purpose, and for liquid culture, except the agar,
And
all other components.
you didn't face any difficulty to cultivate this endophyte.
Ma'am, actually, contamination was a big problem. So, first at the first time when I isolated, when I used these plants for isolation, actually contamination was a problem, and in control plates I could observe some of the colonies. So the second time I just extended the time duration that I have applying for the surface sterilization process, and I could get like I could isolate the bacteria easily. Like
Okay,
almost
okay, okay,
within 2 weeks, I could isolate the bacteria.
okay, okay. What was the assay used for cellulase enzyme?
Ma'am, CMC SSA.
Hmm?
CMC SSA. The substrate used was CMC. And
Okay,
DNS was once the CMC was added to the enzyme preparation, like it will release glucose, and after that, the glucose will react with the DNS reagent.
okay,
Okay.
okay. Good, 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 good. Okay. All the
Thank you,
best
ma'am.
for your
Thank you.
future work. Okay.
Thank you. Thank you so much.
Thank you. Now we'll move on to the next candidate. I welcome Sadhana number BOTPG004.
Yes, ma'am. Good afternoon, everyone. And my topic for today is watershed management structures, enhancing the soil quality using GIS and EIA. And the objectives are to perform EIA in the field while we go for the field visit, and also to observe the watershed management structures there, and also to collect the samples and perform certain tests to understand the correlation between the watershed management structures and the soil quality from ridge to valley. And also after that we had to assess what is the effect that is taking place on the soil and how the soil quality is changing. I'll go for the introduction. This is India country and this Telangana state, and this is the district Jangam where I have done my research. Here somewhere is the village, and this is the village Marigadi where I've performed my project. In here, particularly we have chosen this village because of its highly progressive transformation of the unfertile soil completely into the fertile soil, which resulted in a complete change of a land use pattern, which was barren land before, and then it got completely changed to agricultural land. And then also the effective constructions of the watershed management structures we got to observe in this village. So I've chosen this village. There in that village, I had to perform the this environmental impact assessment. So the environmental impact assessment I would define as a study that we do before constructing any development structure. We go to the place and we study the what are the impacts that will cause on the environment, maybe ecologically, environmentally, based on the economy or the social life. So these are the things we check while we go for the environmental impact assessment. Um, this is the basic definition which was given by the IAIA, that is the decision-making support instrument which aims to identify, predict, evaluate, and mitigate the biophysical, social, and all the environmental impacts. And the EIA also includes the interaction with the public. And we have even interacted with the public there in the village. So this is how the environmental impact assessment includes different sciences. Earth science, life science, and the social science. The life science includes the terrestrial and aquatic ecology. The social science includes the landscape, cultural heritage, the human. And in earth sciences, we included all these: air, water, soil, and geology. And these are the steps of EIA, which include screening, scoping, evaluating the impacts. In screening, we initially go and we make a screening of the development project or the development structure we are going to construct in the village, and then we go for the detailed scoping of the development structure, how it will be affecting the environment and all the social aspects. Then we will evaluate its impacts. Then we interact with the people and we make them aware of what are the benefits of the benefits they get by the structure that is being constructed. Then later on, an EIA report will be made, which will be reviewed again, and the decision making will take place, after which an overall environmental audit will be taking place. These are from some of the sites we have visited in the village Marigadi, and these are all the watershed management structures we have seen. Coming to this, what is watershed? What is watershed? Watershed is, watershed can be defined as a surface area from which the rainwater runoff completely drains out into a common point. So this complete area can be defined as a watershed. So watershed management is something which deals with the entire water in the watershed. The watershed management is a management process that seeks the balance between the health, ecology, economy, and the cultural and social conditions. So this is how the watershed management involves the aspects of environment, economy, and society. In the environment, the watershed management includes water quality, biodiversity, pollution. All these aspects will be covered in the watershed management. And coming to the society, the drinking water of the people living over there, and also the waste management of the people living over there, and also the land use patterns will be checked. How the construction process or the development structure which will be constructed affects the drinking water, how it will affect the land use pattern, etc. And also how it will affect the agriculture pattern of the people living there, and how it will affect the transport, forestry, tourism, manufacturing, etc. So all these things include in the watershed management. Keeping in sense all these three aspects, the watershed management will be done. These are the watershed management structures which I have researched on. Staggered trench, stone bunds and rock dams, farm ponds, check dams, feeder channels, percolation tank. I have particularly chosen this image because what I've done research is on from ridge to valley. So we have climbed up to this point actually, and from here we have been looking into all the watershed management structures. The staggered trenches are constructed here on the top, and the stone bunds or rock dams will be constructed to the way down, and in the slope there will be few here the farm ponds will be constructed. And coming down here there will be certain agricultural lands which I'll show you later in the PPT. Here the check dams will be constructed, the feeder channels, that is the water which will be going into the other streams in the village. Feeder channels will be dug there, and the percolation tanks in the lower land, that is in the from ridge to valley. This is the details of various structures. So this is the CCT. CCT will be constructed actually here, if we consider this as a mountain or a hill, around the mountain the CCTs will be constructed. These are how the CCTs will be constructed around the hill. And due to lack of time, I'll just make it short. I'll just show the images. So these are the rock dams which we have seen, and I've shown you the placement where they'll be constructed. These are the farm ponds, the check dams. From the hill, when the water will be flowing down, the runoff flowing down, it will be collected here and later it will be sent down. This is on the slope actually. These are the feeder channels which from the mainstream they are going into the other streams. These are the percolation tanks, and I've collected samples on the hill, on the slope, and in the valley. the sand, loam, and clay soils, and we have done certain experiments in the laboratory for soil texture, conductivity, pH, moisture content, bulk density, and water holding capacity. We have done it. So this is how the collected samples look during the soil texture test. Um, so these are the results. In the hilltop soil, we can see the sand is 77%. There's no clay is very little, 7%, and the silt is 7%, and the clay is 16%. That means very less fertile. And in the agricultural soil, you just don't see the sand. There's only clay. A lot of clay, and 11% of silt. In the check dam soil, that is the lower soil, that is in the valley, the 13% will be the sand, and 20% will be the silt, and the clay will be 67%. That means this is more fertile and this is a bit less fertile. Soil pH. What happens if the soil pH is neutral and acid? When soil pH is neutral, the balance of the H plus and OH ions is maintained, and the nutrient availability will be abundant, due to which the plant growth will be good. But if the soil is acidic, the H plus ions and will be more in content, due to which the nitrate leaching will take place, and that is the nutrients are very less available, due to which the plant will have a stunted growth. So this is how these are the results. The hilltop have 9.44 pH. Almost 3 of them are neutral pH, but the check dam soil is a bit acidic. So the soil moisture contents were very high in the check dam soil. That is because the water is stagnant to some extent at the check dams. And the agriculture soil, 0.37% of soil moisture was found, and the hilltop, very less soil moisture was found, because the water just runs off to the lower lands. This is coming to the soil bulk density. What is bulk density? Bulk density. Yes, ma'am.
For the interruption. Yeah, Dr. Time is over. Please conclude.
Okay, ma'am. This is the result of soil bulk density and the water holding capacities. So these are the results of the EIA. That is, the groundwater level was increased, the surface water and stream flow was increased, soil erosion was reduced, runoff was reduced, and the land use pattern got changed. So these are just images of before the project was constructed and after, in the initial and later stages. This is how the water was utilized, well maintained. These are the images of at the Marigadi village. These are the farm ponds during construction, after construction. So this is the area where the complete barren land got converted into agricultural land beside the CCT at the slope. So that is the conclusion. From the above study, I have understood that the watershed management structures constructed increase the groundwater level, increase the surface water and stream flow, and various other aspects. So that will be very useful while in these times when the floods are more. Thank you.
Thank you, Sadhana. Now the session is open for the discussions.
Any questions from our audience?
Hello, Sadhana.
Yes, ma'am.
Can we relate your studies to flood management?
Yes, ma'am. To some extent, we can, ma'am. Ma'am, am I visible, ma'am?
Not visible. You're audible.
Okay, ma'am.
If it, can you explain how?
Yes, ma'am.
Sadhana, please turn on your camera.
Sorry, ma'am. There was a network issue. Yes, ma'am. Am I visible, ma'am?
Yes.
Yes.
Yes, ma'am. Yes, ma'am. Ma'am, please carry on with the question. I did not get your question, ma'am.
Can we relate your studies to flood management?
Yeah.
Yes, how we can do that?
Relating it to flood management is a very apt thing we can do, because the flood management is all about managing the water, mainly the runoff water. So this is mostly applicable in the villages right now, because this has been taken up by the Manrega. You might have known Mahatma Gandhi National Rural Development Scheme. So they are doing this, and the floods can be mostly managed by this, because these constructions are does not include a lot of budget. It doesn't need a lot of budget. So this a little bit of awareness, this can be done, because it's all digging the soil and making something up with the soil itself. So this can be very effectively used in the flood management.
Okay.
The constructions I have shown, they can be constructed very easily. So
Thank you.
yes, thank you, ma'am.
Sadhana, I would like to ask you one question.
Yes, ma'am.
How long did you take to complete this project work? How did you start this work?
To
Was it
complete
with collaboration with some agency or some I would like to know about that.
Yes, ma'am. This work I have completed in 3 months, ma'am. This was my PG work. So 3 months I took, but only for the information purpose. I had to collaborate with Rural Development, DRDO, District Rural Development Office of Jangam District, Telangana State. And for the information on the maps and all, I took the help of that department, and I took 3 months for this. The initiation, the thought, the main important thing why I worked on this was, as you mentioned, the water which is wasted most of the times. We only know about the rainwater harvest pit and nothing else. We are much aware of how to utilize this water. So I worked on this village, Marigadi, and I went over there and I looked into it for 3 months, ma'am. It wasn't into collaboration, but for certain information and details, I was in contact with the District Rural Development Office of Kanpur District, Jannam.
Okay, thank you, Salma.
Thank you, ma'am.
Okay, your work is very relevant in today's context.
Thank you so much.
Regarding the EIA draft, new EIA draft, what is your opinion about this EIA 2020 draft?
Any environmentalist, I guess, would oppose the EIA 2020 draft.
Have you responded?
Not really, ma'am, but I had some internal feeling that things should be in a right way, because unless and until you're not disclosing the information to the public won't be aware of anything which is going on in the anything which is being taken up by the government. So unless and until people don't interact, people don't know about the structures the government is initiating or developing, people wouldn't be able to give their opinions, and that would be a one-sided decision according to me, because that from the government point of view, as a government employee or as a bureaucrat, things may be different, but for public, things may be different. So I think a few things should be open for public regarding the 2020 draft.
Okay. Regarding your project, how many sites you have visited? How many watershed you have visited? And what was the size of the watershed?
Size of the watershed? Actually, I visited it last year, ma'am. So I visited it before the during the actual construction. The main village I visited was Marigadi, but I have even visited the surrounding villages like Raghavpur, Shagal, and all. But they were more effective. Like, I just wanted to study it on from ridge to valley. So I found in Marigadi district that was very well constructed. The things were very well constructed in order, as I showed in the PPT, and also the constructions were very
big one. Was it a big one?
Very big one, ma'am. Trust me, the land which was barren, it got completely changed to an agricultural land. That was a pure miracle, I could see, because as an environmentalist, that would be the most happiest thing for me. So I thought, why shouldn't I take up this village itself for the project?
Okay. Then about the sample collection, how did you collected these soil samples from various locations?
Sample collection, actually in Marigadi, as I've mentioned, we went on to the hilltop, and we have checked on the staggered trenches, and from the staggered trenches, near to the staggered trenches, I have collected the soil which was very sandy. And while coming down in the agricultural land which got converted, the barren land which got converted to agriculture, from that land itself I have collected the agricultural soil that is on the slope. And in the valley exactly, there was the check dam construction going on. So from that check dam, I collected the soil, because the water runoff, it will be coming from the ridge to valley, and the soil which is coming down, it brings up all the fertile soil to the check dam. Because you know that soil erosion thing takes place, and the check dam has a very soft kind of soil. So I've collected that soil sample. So I conducted my test on that 3 samples.
Okay, and then what was your contribution in soil analysis?
In soil analysis, ma'am, I've done various tests like pH test and the soil bulk density, water holding capacity.
And you didn't find any difference in the pH?
No, ma'am. There was no much difference in the pH, but in the hilltop soil, the pH was low. That means it was acidic to some extent, I guess. And the plant growth was also very low on the hilltop. So that was the only thing I've seen.
Have you checked the microbial content of the soil?
No, ma'am. Actually, the microbial part I did not go into.
Okay.
But I
Okay.
wish I could do it because my bachelor's was from biotechnology, genetics, so biochemistry, these things, the microbial thing, all of it involves in that. So I wish to do it, but as I'm from Department of Environmental Science, I couldn't do it. I could cover only this
Okay.
part.
Okay. 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 Okay.
Thank you so much, ma'am.
Thank you, Sadhana. Thank you, ma'am.
Thank you so much, ma'am.
I now invite Tansila S, number BOT PG005, to present her project work. And also, participants, they are requested to switch on their videos while presenting. Please switch on your video.
Okay. Good afternoon to one and all. I am Tansila Sadiq. Today, I am standing here before you to present my MSc project on the topic isolation, molecular characterization, and evaluation of plant growth promoting efficacy of endophytic bacteria from Calotropis procera. We know that endophytic bacteria are non-pathogenic partners of plants. In plant-microbe interaction, plants provide nutrients and a residency for bacteria, which in exchange can be directly or indirectly improve plant growth. The direct plant growth promotion can be achieved by phytohormone synthesis and biological nitrogen fixation, making nutrients like phosphate, potassium, etc. indirectly due to their antagonistic activity against plant pathogens and the production of enzymes. നമുക്കറിയാം പ്ലാന്റിന്റെ ഡെവലപ്മെന്റ് ഏറ്റവും അത്യന്താപേക്ഷികമായിട്ടുള്ള ന്യൂട്രിയൻസ് ആണ് നൈട്രജൻ, ഫോസ്ഫറസ്, പൊട്ടാസ്യം. ഈ ന്യൂട്രിയൻസ് എൻഡോഫൈറ്റിക് ബാക്ടീരിയാസിനെ പ്രൊഡ്യൂസ് ചെയ്യാനുള്ള
Excuse me, Tansila. I request you to kindly present in English itself because we have participants from outside Kerala as well.
Okay. Almost 3 lakh plant species that exist on our planet. Only a few of these plants have been completely tested for the presence of endophytic bacteria within their tissue. Calotropis procera is one of the medicinally important plants which containing a wide variety of secondary metabolites. These can be used as medicine for various diseases. So, Calotropis procera is selected for the study. The objective, the isolation of endophytic bacteria from Calotropis procera to study the effect of endophyte on growth Calotropis procera, biochemical and molecular characterization of selected endophytic bacteria. Hypothesis. Endophytic bacteria, endophytic species present in Calotropis help enhance plant growth by the secretion of plant growth promoters or shows enzyme production which add for better growth. Methodology. Endophytes are isolated by initial surface sterilization, followed by culturing from ground tissue extract or nutrient agar medium by spread plate method, followed by subculturing by streak plate method. On the basis of morphological and phenotypic characters, 10 isolates are selected for further study. We know that endophytes are increased plant growth directly or indirectly. Direct growth promotion was done by IAA production, phosphate solubilization, nitrogen fixation, and potassium solubilization. In IAA production assay was done by test bacterial culture was inoculated in the nutrient broth with a tryptophan incubated at 30 degrees Celsius for 48 hours. Culture was centrifuged at 3000 rpm for 30 minutes. Then supernatant was collected and 2 drops of orthophosphoric acid and 4 ml of Salkovsky reagent was added. The pink color which detect the presence of IAA. The phosphate solubilization was done by all the isolates were inoculated into the 100 ml of nutrient broth and incubated at the room temperature for 24 hours. Then it was added to the center of pKa medium, the halo zone which indicates the presence of phosphate solubilization. Nitrogen fixation was done in the Ashby's mannitol agar plate. Potassium solubilization was done by Bell diffusion method in Alexandro agar medium. Out of 10 isolates, 5 shows IAA production and 2 shows in nitrogen fixing bacteria and 1 shows potassium solubilizing bacteria. Enzyme activity was done by in 3 tests: amylase test, protease test, and lipase test. Amylase test was done in starch agar medium, and protease test was done in milk agar medium, and lipase test 100 ml of nutrient agar was prepared and 1 ml of 220 added to it, and it is then autoclaved. The bacterial was inoculated into it and incubated. Then clear zone which shows lipase activity. In this enzyme activity, out of 10 isolate, 5 shows lipase activity and other tests are negative. In this study, antimicrobial activity was done that can be detected in 5 test organisms like Bacillus subtilis, Bacillus cereus, Staphylococcus aureus, and E. coli. And out of 10 isolate, 4 shows antimicrobial activity. The third isolate shows higher antibacterial and the second most plant growth promoting activity. It's selected for the further biochemical and molecular identification. After the biochemical of the 8 sample, which shows gram positive cocktail with a positive result on TSI, citrate, and catalase. In molecular characterization, isolation of bacterial DNA by using CETA method after the isolation of genomic DNA that can be purified by agarose-gel electrophoresis and amplified by using PCR. The DNA sequence were analyzed by using BLAST-N rule. After the BLAST search result showed a similarity of 99.89% over various strains of Staphylococcus hominis. It has an E value with 0.0. This is the BLAST tree view image, shows the relationship of various strains of Staphylococcus hominis. The bacterial strain number 8 shows a considerable reduction in IAA, nitrogen fixation, and potassium solubilization. Sequencing of 16S rRNA and BLAST analysis confirm the strains is Staphylococcus hominis. We concluded that the results show the bacterial strain Staphylococcus hominis help in enhancing the plant growth in plant. This gives a hope in agriculture to develop biofertilizer that are profitable, environmentally sound, and good for communities. In future perspective, develop a potent biofertilizer by manipulating the genome of Staphylococcus hominis. Thank you.
Any questions from audience?
Tansila,
Yes, Miss.
what were the morphological features that help you to identify this bacterial strain?
Color, morphology, elevation, etc.
What? Morphological features that you have observed in the culture plate?
Colony color, shape, elevation.
Can you explain its shape?
Shape, irregular and circular shaped bacteria can be obtained. Colony color, off-white color, yellow, light orange, white are obtained.
Okay, Tansila, thank you. Thank you so much.
Tansila, I have one question. Apart from this Staphylococcus bacteria, which are the other strains, other bacteria that we usually find as endophytic bacteria?
Ma'am, the 8th sample which shows higher antimicrobial and plant growth promoting property. So sample 8 is selected for the biochemical and molecular characterization, and other bacteria are.
Okay, one more question. You showed an amplified DNA in one of these slides. So which product was actually used?
Ma'am, is it clear? Hello?
Amplified DNA agent. Can you hear me?
Nisha ma'am, there is some net issue. You are not
not audible?
Yeah.
Okay. Am I audible now?
Yes, yes.
Yes, okay. Tansila, what I asked was, in one of the slides you have shown an amplified DNA. So which primer have you used for that?
Forward primer 20F and a reverse primer 1429.
Hello, I would like to know which gene actually you intended to know. Which gene have you tried to amplify?
16 SRRN.
Okay, okay, fine. Thank you.
Thank you.
Okay, Tansila, you have sequenced those bacteria and have you deposited in GenBank or?
Yes, ma'am.
Yes. How many species you got?
Morphological and phenotypic characterization, I select 10 bacteria.
You selected 10 and all of the 10 you have sequenced also?
No, ma'am. The growth plant growth promoting bacteria is selected for the study. Molecular and biochemical characterization.
Okay, how did you check the plant growth promoting factors? What all parameters you have taken?
Nitrogen, phosphorus, potassium is one of the most important nutrients for plant growth. So,
okay. How did you analyze the phosphate solubility?
Phosphate solubilization.
You are not visible.
Next correct കിട്ടില്ല മിസ് അതുകൊണ്ടാ.
Okay, okay.
Phosphate solubilization, the bacteria was inoculated into the nutrient broth and then it was added to the pK medium.
What was
halozone.
What was that medium?
Hello,
what was that medium? Is there any full form of that?
Yes, ma'am. I forget it.
Okay. Okay, continue.
pK medium. All the plates are incubated at the room temperature, and then halozone, which shows halozone around the colony, which shows the positive result.
You have used a ready-made medium, or you have prepared the whole medium with the minerals?
Ready-made medium.
Okay, okay. Then you have done blast, and what was the result you got?
That shows 99.89% similarity with the Staphylococcus hominis.
Okay. Okay, good.
Good afternoon, everyone. And so I'm Bhanupriya. I would like to present my MSc dissertation, a comparative micromorpho-anatomical studies of the genus Lygodium from Kerala. So let me introduce Lygodium. Lygodium is a terrestrial fern with slender stem alternately pinnate, climbing by twining branches. And it belongs to the family Lygodiaceae under PPG 1 2016 classification. And the peculiar feature of Lygodium is the presence of sporangia forming monosporangial sauroid. And from Kerala, only 2 species were reported, that are Lygodium flexosum and Lygodium microphyllum by Kerala Forest Research Institute in 1998. And also it is published in Handbook of Fern and Fern Allied of Kerala in 2015 by P.V. Madhusudanan. So moving to the issues that is relating with the data existing now, you can see there is no proper classification system of the genus Lygodium. So even though the existing classification system used only 1 or 2 morphological feature for its comparison, and also there is no proper study of this species from India. So to resolve these issues, I am moving to my objective, that is to identify Lygodium species based on leaf morphology, then to study the anatomical variations, including the anatomy of rhizome, root, petiole, rachis, and stomatal variations, then to determine the germination period of the spore and also the pattern of gametophyte development, then to determine the spore morphology, number and structure of sporangium, shape of indusium, length of saurophore among the species, And finally, to approximate the sequence level variation using tRNL sequence. So moving to the methodology, first I collected the different species, then from different sites of Kerala, then morphological study were done, and then anatomical study were taken by hand sectioning, and its photographs were taken using mobile phone, and then spore analysis, including the SEM analysis is done. Then its gametophytic pattern of growth and its period of germination were observed, and finally the in silico analysis were done. So moving to the result and discussion, and my first objective is to identify Lygodium species based on leaf morphology. You can see that here in the in Lygodium flexosum, the pinnule is lobed, and also the midrib is prominent in Lygodium flexosum. Whereas in Lygodium in microphyllum, you can see that the pinnules are not lobed, and also the midrib is not prominent in case of Lygodium microphyllum when you compare the flexosum and microphyllum by leaf morphology. So moving to the anatomical variation, and you can see in Lygodium flexosum, where in Rachis anatomy you can see the prominent lateral wings are seen in flexosum, but it is absent in the case of microphyllum. And also, if you observe the vascular bundles, the T-shaped or V-shaped xylem can be seen in the flexosum, whereas in microphyllum, the xylem occupied as a solid central mass in microphyllum. But coming to the petiole anatomy, there is a unique character that can be seen in Lygodium flexosum. There is a green chloroplast cells can be seen inside the vascular bundles, which is a unique feature that can be seen only in Lygodium flexosum. But coming to microphyllum, such chloroplast cells are absent in the microphyllum. But the cortex region will be completely filled with the sclerids in case of microphyllum, but in flexosum it is of parenchymatous. Then in rhizome anatomy, both the features are similar in flexosum and microphyllum. Only one thing that we can differentiate them is the presence of sclerids. So tannin cells can be seen in Lygodium microphyllum, but in flexosum it is absent. In the root anatomy, in the inner cortex of Lygodium flexosum, you could see 2 to 4 layers of sclerids in flexosum, but the sclerids are absent in Lygodium microphyllum. In the root anatomy, only you can see it is parenchymatous, which is filled with the starch granules. Then another observation that I made while taking the anatomical section of rachis in Lygodium flexosum, that is in the base of the rachis, it is usually and the xylem occupies most of the vascular bundles, but its xylem shows variation with increasing distance from the base of the rachis to the middle region. In the middle region, a prominent T-shaped structure can be seen, and also the phloem is also much more. You can observe it in the middle region of the rachis. Then in the stomata, in the flexosum, it is anemocytic, whereas in Lygodium microphyllum, it is diacytic. Then, to determine the germination period of the spore of both the Lygodium flexosum and microphyllum, I given a similar condition to the flexosum and microphyllum. Then I could observe that the flexosum, the germination of the spore take place within 6 days, and you can see as a heart-shaped prothallus, and also it is symmetrical. Whereas coming to microphyllum, the germination rate was too low, and also it takes 3 months for the germination of the spore. And when you observe the prothallus, it is asymmetrical. One short wing and a large wing can be seen in the microphyllum, whereas it is symmetrical in case of flexosum. And moving to the fourth objective, to determine the spore morphology. If you observe the sorophore microscopic, I took the sorophore microscopic view. Then in Lygodium flexosum, the sporangia is closely placed, whereas in microphyllum, you can see the sporangia is raised, and also we can clearly distinguish each sporangia. But in case of then moving to the pseudo-indusium, this indusium, the margin of the indusium in Lygodium flexosum, it is not much wavy or any lobing cannot be seen in the Lygodium flexosum, but in microphyllum, a clear wavy or lobed nature can be seen in the Lygodium microphyllum. Then the spore morphology. The spore in Flexosum is trilate, isosporous and triangular, which is pale yellow colored. But in coming to Lygodium microphyllum, which is subtriangular to circular, and it is also isosporous. And then I go for taking the SEM analysis. Then I could see that in Lygodium flexosum, the verrucate ornamentation is clear, and also the presence of spheroids can be seen in the surface of the spore. But in microphyllum, it is a reticulate venation, and there is no spheroids can be seen at the surface. Then in the fifth objective is going for the in silico analysis using tRNL sequence. I took retrieve the sequences from NZBI, and then I observed its variations. Then we can see that in Lygodium flexosum and microphyllum are clearly or distinctly placed in different clades. But then I move for the sequence alignment of the Lygodium flexosum and microphyllum. I could found 62 different sites are there. 62 different nucleotide sites can be seen. So these 62 different nucleotides are giving this Lygodium flexosum and microphyllum unique features or separating these two species. So let's conclude that the study showed the two species, Lygodium flexosum and microphyllum, can be clearly differentiated based on its morpho-anatomical and spore features. And this is the first report on the anatomical study of Lygodium microphyllum. And also this work report the anatomy of the rhizome, root and petiole of the Lygodium flexosum for the first time. So the current study is useful for the systematics of the genus as well as for the phylogenetic purpose. So I submit a complete catalogue of this species from Kerala. Thank you.
Thank you, Bhanupriya. The session is now open for the discussions.
Yeah, Bhanupriya,
Yes,
where all the pictures in the PPT, the pictures you have presented, were all of them clicked by you?
Yes, ma'am. Yes, I do.
The microscopic pictures also.
Yes, microscopic pictures.
Yeah, it's really nice.
Thank you, ma'am. Thank you so much.
Bhanupriya,
Yes, ma'am.
nice presentation. I want to know the methodology for germination studies. You have told that you have conducted germination studies.
Yeah. I given
Can
the
you explain
natural condition itself. I took the two petri plates and take the soil and giving a sterile condition, and the same condition given for both the spores. Just a natural condition itself I given, but then I observe because within a limited time I can't go. Even I tried tissue culture also, tissue culture technique for the growth of germination of the spore, but it doesn't work with microfilm. Then I given the similar condition and such natural condition itself. Then I observe the growth pattern, germination period and all.
Have you done any sterilization procedure?
Yeah, the soil was just heat dried and placed in the petri plate, and it's in the normal condition itself.
Okay. Thank you.
Thank you.
Okay, you have compared the two species of Lygodium,
Yes.
and what was your conclusion? Whether the similarity was more or whether the differences were more?
Yeah, I try to my aim or my hypothesis is to to check other than morphological feature. In the existing data, we can see the classification of Lygodium flexosum and microphyllum is made only by morphological feature. But through my study, I could find that this flexosum and microphyllum is showing a lot of ecotypic variation in its leaf length, pinnules, lobing pattern, and all. So I need to go, check what are the anatomical variation or spore variations that can used to classify these two species. Because from India, mainly its ethnomedicinal properties are checking. The n number of data are available from the literature regarding its ethnomedicinal importance. So there is always a chance for getting difficulty in finding these two species. So I tried for its anatomical variation and spore variations can be used for differentiating these two species. If you check the anatomical variation, we can clearly differentiate these two species.
Okay. You have mentioned there is ecotypical variation.
Yes,
so,
ma'am.
How did you collected the sample? How many samples you were collected?
I collected these two
And
species.
the location
Different sites from Kerala, that is from Kottayam, Ernakulam, then Thiruvananthapuram, then from here Wayanad, Kasaragod, and from all the districts. Mostly I covered all the districts except from Kollam.
How
And
many samples from each district?
3 samples from each district.
Okay. Then you can include some statistical analysis also.
Yes, ma'am. I think it's a coming to a lengthy.
Okay. But it's a must.
Yeah, okay, ma'am. Okay, definitely. I will include.
Okay. Then I must ask, what is the significance for comparing these two?
Yeah, that is no one has tried the anatomical variations, and as per my observation, my literature review. When even the Lygodium flexosum is usually used in my home also for as a traditional medicinal system for wound healing. So just I go for checking whether any other species are existing in Kerala. Then I found that there is no such works regarding its anatomy and all. So that I go for so it will be an.
Does it have any ethnomedical importance?
Yes, lot of ethnomedical importance, because it is using mainly and now the research is going regarding in the presence of ligodinolide content in Lygodium flexosum. It is not completed. They just reported the presence of ligodinolide content in literature reviews and all. And so which is wound healing property, and also which is used in case of gastro issues and all. Flexosum and microfilum is widely using.
Okay, thank you.
Thank you, ma'am.
Thank you, ma'am. Thank you, Bhanupriya. Now I invite Arya AR to present her project. Arya AR?
Yes, ma'am. Is it visible, ma'am?
Yes, it is.
Yes.
Good afternoon, everyone. I'm Arya. Today I want to discuss about my topic on carbon dioxide sequestration studies as a sustainability initiative. We all know today in our world, climate change is one of the core issues and has lots of impacts on our ecology. Since from industrial revolution, the fossil fuel consumption has increased with the carbon emissions, which is the main root of global warming and climatic change. In the present time, carbon emissions are being proxy for environmental degradation. Like there is a solution for every issue, trees have major role in mitigating tremendous release of carbon dioxide into the atmosphere through so-called process called carbon sequestration. It is the process by which the atmospheric carbon dioxide is taken up by trees, grasses and other plants through photosynthesis and stored carbon as biomass. Let's look into the objectives of my work. To count the number of trees in the campus and measurement of their height, GBH. GBH means girth at breast height. To calculate the AGB. AGB means above ground biomass. It is the biomass above the surface of the soil. BGB. BGB means below ground biomass. It is the biomass below the ground level. Total biomass is the sum of the AGB and BGB. To calculate the average sequestration of tree species within the campus, to classify the plant species for their carbon dioxide sequestration efficiency, to identify the plant species which can respond with highest efficiency to change in terms of high carbon sink capacity, as a mitigation strategy of carbon. Now let's move on to the materials and methods. Counting of individual tree species in the campus, estimation of tree height using clinometer, and calculation according to the formula. Tree height is equal to h plus d into tan A. This is the formula used for calculation. Measurement of GBH using a tap. Assessment of wood density of each species from a database called ICRAF database. Calculation of AGB, BGB, and total biomass using standard equation. To calculate AGB, I used the equation volume of tree into wood density. BGB can be calculated using the formula AGB into 0.26. Total biomass is the sum of AGB and BGB. Carbon store can be estimated using the formula total biomass divided by 2. The average amount of carbon sequestered can be calculated by the formula carbon store into 3.67. Also, I done several statistical analysis of data using Excel state software. Then we have to look into the 4 steps for measuring the carbon dioxide sequestered in a tree. Estimation of total height of tree using the GBH of the trunk and an estimate of the height. Conversion of weight of the tree to the dry weight of organic matter. Estimation of amount of carbon being a proportion of dry weight. Next, the conversion of amount of carbon to the equivalent amount of carbon dioxide sequestered. Let's move on to the most important section, the results. Present study observed considerable diversity of tree species within the campus. The total number of trees in the campus was counted to be 253, belonging to 52 species distributed in 30 families as per APG system. Study includes families such as Fabaceae, Ericaceae, Lauraceae, Euphorbiaceae, Mithraceae, Lespidiaceae, Rubiaceae, and so on, and etc., etc., etc. Let's move on to the graphical representations. These graphs are constructed using the Excel state software with its new version, trial version. These are the diagrams made by putting the parameters in its decreasing order. In figure 1, distributing the tree heights in this Carelia brachiata is the most highest member belonging to the Rhizosphoraceae family, followed by Tamarindus indica and Anacardium occidentale. In figure 2, distributing the GBH, Tamarindus indica exhibit the highest value, and least GBH was recorded in Flecosia gengomas, belonging to family Bixaceae. In figure 3, show the distribution of total biomass. Here also Tamarindus indica exhibit the highest total biomass, and least by Podium variegatum, belongs to family Euphorbiaceae. In figure 4, shows the distribution of average amount of carbon dioxide sequestered by different plant species. Here also Tamarindus indica exhibit the highest value and least in Podium variegatum. In figure 5, shows the distribution of average amount of carbon dioxide sequestered per plant families in the campus. Here Fabaceae. You can see its Fabaceae exhibit the highest value and Poaceae exhibit the least one. In final, in figure 6 is a pie chart showing the carbon dioxide sequestration potential of angiosperm families within the campus, and Fabaceae has the highest carbon dioxide sequestration capacity. Next is a table showing the average amount of carbon dioxide sequestered per plant families. We can see Fabaceae sequestered about 1,39,064 kilogram of carbon dioxide. Let me check into the correlation analysis of this parameter. It was performed using correlation analysis was performed using Excel state software. This is a trial version for 14 days with the help of my case. Evaluated the strength of correlation between tree height, GBH, total biomass, and amount of carbon dioxide sequestered. We all know in a correlation analysis, 0 indicates no correlation, 0 to 1 indicates positive correlation, and -1 to 0 indicates negative correlation. In figure 7, you can see shows the perfective a perfect positive correlation between the plant biomass and carbon dioxide sequestered with the correlation coefficient of 0.9721. In figure 8 gives a strong positive correlation between the GBH and the carbon dioxide sequestered with a correlation coefficient of 0.7526. In figure 9 gives a moderate correlation between the tree height and the carbon dioxide sequestration with a correlation coefficient of 0.6103. In figure 10, it reveals moderate correlation between the tree height and total biomass, while positive correlation exists between the GBH and total biomass. Let's sum up. The total number of trees are 253, belongs to 52 species distributed in 30 families as per APT4. Major tree species are Cocos nucifera, Sweetenia, Macrophylla, Peltrophorum, Herocarpum, Polyalthia longifolia, Tectongandis, like this. Fabaceae found to be the dominant family. Carelia brachiata recorded the maximum height and leaves in Podium variegatum. Tamarindus indicus exhibit the highest GBH value. Wood density was least in Carelia brachiata and highest in Cisalpinea pulcherrima. Similar to height, Podium variegatum exhibit the lowest GBH, AGB, DGB and total biomass. The Tamarindus exhibit the highest values of all these parameters. Tamarindus indica exhibit the highest carbon dioxide sequestration, which can respond efficiently to climate change in terms of the carbon mitigation strategy in the urbanized world. Thank you.
Okay, thank you, Arya. The session is open for discussion.
Arya,
Yes, ma'am.
can you explain about why this Tamarindus indicus will be having the highest carbon dioxide sequestration capacity according to the physiological view? Why it will be having?
Yes, ma'am. Yes. Tamarindus indica has the highest GBH value, has high biomass as compared to other members. Biomass is more to Cisalpinea pulcherrima, but but Tamarindus indicus also have high wood density. Biomass, അതുപോലെ തന്നെ AGB, BGB also. All parameters are highest for Tamarindus indicus. GBH is about 167, I think. 312 centimeters. It is higher. GBH is very highest for Tamarindus indica.
So these are the morphological reasons. What will be the physiological reason for this? Anything about its pathway or something like that?
I didn't check, ma'am. I didn't extend my study for that.
Okay. Okay, thank you very much.
Okay.
Okay, Arya, you have explored your campus, and how many trees?
It's about 253 trees.
You are mainly focused on your work is mainly focused on trees or
trees, only trees, ma'am.
Only trees. Okay. So, is there any variation in height?
Yes, very many variations, ma'am. It's about 81 meter to 3 meter. Very high variations in height.
And what was your finding? Does
My
height have any impact?
Yes, height. The height has impact like other parameters.
And the age of the plant, have you considered?
I have, due to the issues in the present day, I could not collect about the age of the tree. So I stopped my work in average carbon dioxide sequestered within the campus, with the trees. I couldn't collect data about the ages of trees.
Okay, okay. So what was your conclusion?
Conclusion, Tamarindus indica the trees belonging to Fabaceae mainly exhibit more carbon
What all trees? What all trees you have analyzed in Fabaceae?
Fabaceae, I think Peltophorum therocarpum, Albizia salmon, um, for Tamarindus indica, Cisalpania pulcherrima.
Cisalpania pulcherrima is not a big tree.
But in my campus there is a large tree, Cisalpania pulcherrima.
Okay.
It has a wood density of 0.95, ma'am. It has a large carbon dioxide sequestration capability.
Did you find any bamboo in the campus?
Yes. I
And
recorded the rate of
impact of bamboo.
It has very low carbon dioxide sequestration capacity. It has very.
But I have heard that bamboo is very good in carbon sequestration.
But in my campus, bamboo is very short and very thin.
Okay, then okay, how much big was that? Bamboo?
It's about 10 centimeter in GBH and about
that size.
That size.
That's right.
Yes.
Okay. Then, so your conclusion is that we have to plant more tamarindus.
Not it. We have to plant more large trees. With more biomass, GBH.
Apart from tamarindus, which plant was the next higher to that?
Macaranga peltata, belonging to Euphobaceae family. Sweetenia macrophylla belongs to Meliaceae family. Tectona grandis belong to Lamiaceae family.
Okay.
Yes, ma'am. Thank you.
Okay, good.
Thank you, ma'am.
Thank you, ma'am. Thank you, Arya. Now I invite Surabhi SP, number BOT PG009, to present her project work. Surabhi?
Yes, ma'am.
Yes, you can present.
Ma'am, is my PPT visible?
Yes.
Yes.
Okay, shall I start?
Yes, you can.
Hello everyone. A very warm good afternoon to all and all present here. I am Surabhi SB. As part of the completion of my master degree, I have done a project under the supervision of my guide, Dr. Rashmi teacher, on the topic floristic documentation of angiosperm taxa in Kukripara. And as you all know, I am here to present a short summary of what I have done. So let's begin with the background of this work. As we all know, the exploration and documentation of plant diversity is considered as the backbone of biodiversity assessment. In all point of views, especially the medicinal, economic and ecological aspect, we have to document the details of plants prevailing at a particular area in order to find out how diverse the area is, or is there any need for conservation in that area. If we check previous data available on the floristic studies, it can be found that numerous works have been done to explore the floristic diversity of different parts of Trivandrum district. However, we can also notice that most of the eastern region of the district are yet to be explored or to be documented. On considering this scenario, I have decided to select an unexplored area, Marnallur Panchayat of Katakada Taluk. Since the area is currently unexplored, the study have been attempted to record the floristic wealth, especially angiosperm, in order to assess the human impact on the area and also to take necessary action for the conservation of the area if needed. The present study was designed with these major objectives: multiple field surveys to list out and collect plants from the study area, preparation of herbaria of the collected plant specimen, preparation of artificial keys for the collected plants and their identification, arrangement of taxa as per Bandhaman Hooker system, and its systematic enumeration and description, documentation of endemic, economically and medicinally important plants in the area, assessment of conservation status of the document taxa, documentation of major invasive weeds in the area, and also assessment of the impact of human activities in the flora of the study site. Then comes to the materials and methods. As told earlier, the study area I have chosen is Kukkiripara in Marnallur Panchayat, which is currently unexplored, spread in an area of 1.65 acres of land, and it is the proposed site of biopark by Kerala Biodiversity Board. However, the area has shown to undergo severe environmental degradation as part of construction activities, and also as evidenced by the presence of 3 abandoned quarry sites. As you can see, the first picture shows the electric crematorium in the study area, and second one is the abandoned quarry site prevailing there. As it is a field study, the major materials used are listed here. Major methodologies adopted to do this work include pedestrian surveys, which was conducted in the time period of 1 year in order to understand the floristic diversity, especially to note down the flowering season of the plants. Most of the plants were identified in the field itself and were recorded in the field book instantly, and obviously the unidentified plants were collected and critically observed and dissected for their proper identification. The plant specimens were collected for herbarium preparation and was prepared as per standard specifications. An artificial diagnostic key is also being prepared based on the standard flora. Then the endemism and conservation status of the listed specimens have been evaluated using NVIS database and other previous authentic literatures. As part of this project, during the field trips, semi-structured interviews were conducted with the old peoples living there for collecting traditional knowledge about the ethnobotanically important plants. Photographs of almost all plant specimens were taken from the area. Now, moving on to the result I got from the study. This is the artificial diagnostic key I have been prepared for the easy identification of plants prevailing in the study area. A detailed description of each and every plant were noted down as per the model of a standard flora. The table shows the distribution of angiosperm diversity in the study area. Here, a total of 37 families were recorded, of which 33 were dicot and 4 were monocot. Of the 99 genus found, 81 were dicot and 18 were monocot. And broadly said, about 110 species of plants are present in the study area, of which 92 were dicot and only 19 species are represented by monocots. Some of the rare plants I have found there were Hugonia mystax, Echinocarpus frutescens, Electris isthora, Valtria indica, Smilax, etc. This is the list of most represented families documented from the study area. It shows that Leguminosae represent the most abundant family with 17 plant species, then Poaceae with 12 species, Euphorbia 10, and so on. This is the graphical representation of which shows the percentage distribution of families, and it is clear that the largest family, Leguminosae, represent about 30% of the total angiosperm flora. Then about 21% is represented by Poaceae. Now it is evident that about 50% of total plant documented were belong to these two families. This table shows the habit diversity. Here we can say that most of the plants are herbaceous and represented by 49 species. Then 25 were trees, 17 shrubs and climbers each, and only 2 were creepers. The one and only monocot climber found there was Smilax, which is a monocot plant with reticulate venation. Nine plants were documented, which shows ethnomedicinal values. These plants were listed by conducting semi-structured interviews with the old people residing in the area. Mainly the used part as well as usage details were noted. This is the list of endemic species in the study area. A total of 7 plants have been included, and this list was prepared with the help of NVIS database and other previous literatures. The major invasive weeds spreading in the area include Purpurea passiolis, Mecania scandens, Epitorium odoratum, and Cuscuta reflexa. On completing this documentation, the following conclusions are obtained. It has been found that 110 species of angiosperm belonging to 99 genera and 37 families were recorded, of which 91 species with 33 families were dicot and remaining 19 species with 4 families were represented by monocots. Largest monocot family by Poaceae with 12 species. Leguminosae was found to be the most diverse family with respect to the habit, with herbs 6 species, trees 5, climbers 4, and creepers 2 species. On an average, most of the plants were herbaceous. A total of 15 plants were edible and 11 of them shows economic value. 29 plants were used as ethnomedicine by the local peoples. Then a total of 7 plants were found to be endemic and also 4 major invasive weeds were documented in the present study. Thus, it has been concluded that Kukripara shows only moderate diversity and endemism, mainly due to the human activities and spreading weeds. So, due consideration and innovative conservation program have to be implemented to enhance the species richness of the study area. These are the photographs I have took from the study area. Thank you.
Thank you, Surabhi. The session is now open for discussion.
Surabhi, I just want to ask one question.
Yes, ma'am.
Is anyone helped you to identify the plants?
Yes, ma'am. Actually, our HOD of our college, actually she was retired now, but she helped a lot in identifying the plant species.
Okay, thank you.
Okay, Surabhi, what was the authentic book you have referred for identifying these plants?
Mainly the flora of Presidency of Madras, and also Trivandrum flora is also referred. But most of the plants are identified by. Yes, ma'am.
You have identified it yourself?
Not actually our previous HOD. I told that.
Okay.
She helped a lot.
Okay. You are presenting a floristic documentation, so more pictures was needed. You have given those photos, but it is much smaller size. It is not visible actually.
I say about 82 plants were photographed
and
too much crowded, so I reduced the size.
Okay, you yourself took these pictures.
Sorry, ma'am?
You yourself took this picture?
Sorry, ma'am.
All these photos, you have captured it yourself?
Yes, me and my friend from my college.
Okay. Then, what was your observation? In general, in a summer, to sum up, what was your observation?
Actually, the present area shows only moderate diversity. I think mainly due to the human invasion in this area. And
what will be the impact of that quarry nearby?
Yes, there are about 3 abandoned quarry sites there, and due to all this, it will automatically affect the vegetation in these areas.
Is there any previous data to compare?
No. Kukripara is currently unexplored and no previous data is available.
Okay. You have analyzed a lot of plants, and is there any proportional variation in the indigenous, exotic and invasive plants? You have mentioned invasive.
Yes, I already mentioned the important invasive weeds.
What were the invasive ones?
Invasive means
what were the
plants which
invasive
are brought
plants?
from another area, not indigenous.
What all species you find as invasive?
Epitore moderatum, Mecania scandens, etc.
Okay. Was it an exotic?
Mecania scandens is an exotic variety. It is one of the top 10 weed, I think.
What was the percentage you haven't analyzed?
Actually, the diversity index was not calculated due to the present conditions.
Okay. I have a suggestion. You have done a floristic documentation, and nowadays it is important to digitalize all this. You
Yes,
can
ma'am.
take that step also.
Actually, key is prepared and a key diagnostic key is prepared.
You have prepared a key. That's good.
Yes. Complete description of the whole plant is detailedly described.
Okay. Okay. Thank you.
Thank you, ma'am. Thank you, Surabhi. I now invite Jessina T, number BOT UG0010, to present her project work.
Hello, ma'am.
Hello, Jessina.
Hello, am I audible, ma'am?
Yes, you are.
Okay, ma'am. Okay, a very warm good evening to everyone. My name is Jessina, and here I'm accompanied with my teammates, Miss Anupama Shiji and Miss Nirja Murli. We are all third year BSc microbiology students. So, okay, since we have only 8 minutes, let us quickly go to the presentation. So here's my topic. The topic is Gliricidia sapium as a biological tool for the eradication of Eichhornia crassus. So let us look at the introduction about Eichhornia. So, as we all know, Eichhornia is an aquatic flowering plant. It is a native to South America, later spread to different countries and their water bodies. It is commonly called as water hyacinth, and it grows in wetlands, rivers, etc. They are able to tolerate water level fluctuations, and it is an invasive aquatic plant. That is, they form thick layer over the surface of the water bodies, and it is named as Terror of Bengal as it grows and spreads in an alarming manner. So now let us look at the problems caused by Eichhornia. Eichhornia causes evapotranspiration, that is, it is responsible for water loss, and also the water balance is also affected. It is a major for sedimentation causing flood and soil erosion, and the irrigation and drainage system of the agricultural sector is affected by this plant, and thereby this reduction in source of income. The negative impact on fish transportation, and of course we all know that it is mainly affected in the tourism sectors nowadays, and the water quality is adversely affected. So these are the existing physical, chemical and biological methods for the eradication of Eichhornia from the infested areas. So moving to the physical methods, it includes the manually removing or mechanically removing the Eichhornia from the infested areas by the harvesters or by using chopping machines. Hand pull or hand tool methods are also used here. Heavy machines like bucket cranes, aquatic weed harvesters, etc. are also being used for this purpose. This physical method has certain drawbacks. It is that it is a labor intensive and time consuming work. Also, the heavy engine cost is really high, and also it is not effective in if the infestation is in a large area. Okay, now let us look at the chemical methods. The chemical methods include the use of aquatic herbicide and pesticide. We are all familiar with what will be the side effects if a pesticide is directly introduced into a water body. So 2,4-diqual phosphate and are the main herbicide and pesticide used for this purpose. And as I said earlier, it has also several drawbacks. That is, it causes many environmental side effects. It is an expensive method, and also there requires specially trained persons or special conditions to apply this method, and also it provides only a temporary relief. So if you look at the biological method here, this includes the release of special host-specific arthropods and fungi onto the infested area. These arthropods infect the Eichhornia and finally tend to decompose them.
So weed weevils and nicotinous species are mainly used for this purpose. So it also has certain drawback. That is, the lifespan of these weed weevils is only 90 days. Therefore, there is a need for periodically renewing them or replacing them whenever they are needed. And also, it is not known whether these weed weevils feed on other aquatic life forms or not. Therefore, the success rate is limited. Here, I would like to suggest the use of Glircidia sapiem to eradicate Eichhornia. For that, let us look at what is Glircidia. As we all know, Glircidia is a multi-purpose leguminous tree belonging to the Fabaceae family. They are able to grow on a wide range of soil, and they are useful as a green manure. They have high protein supplement, therefore used for foraging animals. So these are the applications of Glircidia sapiem. It is applied in fields of fodder. It is applied for fodder, firewood. It is used as a green manure. It is used for intercropping. That is, since it is a leguminous plant, farmers use this crop in their fields to increase the fertility of the soil. And it is in countries like Latin America, farmers made paste from these Glircidia sapiem plants to bathe their livestock, since they helps to ward off bot flies or tosalos. And in countries like Philippines, the extract taken from the leaves of these Glircidia sapiem plants was made into an anti-mange dog shampoo. So these are the main properties of Glircidia sapien plant. It is an excellent insect repellent. It has antimicrobial and antifungal property. And it is seen that they are able to inhibit the growth of larvae of Aedes aegypti mosquitoes, which is a kind of mosquito seen more associated with the Eichhornia plant, which causes several infectious diseases like yellow pyrexia, chikungunya, dengue fever, etc. So here I would like to say how we can use Glircidia as a biological tool. We all know that Eichhornia is pollinated mainly through insects and by wind. Since wind is a natural phenomenon, we cannot control it. If their pollinating agents, that is, if the insects number is reduced, we would be able to reduce their sexual reproduction, and thereby we can also obviate several diseases associated with this Eichhornia also. So here is the advantage of using Glircidia sapiem as a biological tool. It is an easily available plant. We are familiar with Glircidia sapiem plant. It is an easily available plant. It is of low cost and low labor is required for their maintenance. They are able to tolerate low soil fertility and are able to grow in almost every environmental conditions. So here I would like to conclude by saying that this project is a completely eco-friendly or environmental friendly method. The eradication of Eichhornia help us to replenish the ecosystem, favoring the magnification or growth of fishes and other aquatic organisms, and helps us in maintaining the natural habitat of the water bodies. Thus, I would like to conclude by saying that this current proposed project is a promising method to build up a healthy, happy and salubrious society. Thank you.
Thank you, Jasina. This session is now open for discussion.
Yes, Jasina. What is the initiation for the growth of water hyacinth or Eichhornia? What is the reason for the initiation of its growth?
See, Eichhornia is mainly pollinated through insects and wind. So it is not pollinated through water. There is a false knowledge that it is mainly pollinated through water, but it is mainly pollinated through insects and through wind. Since wind is a natural phenomenon and we cannot create a vacuum condition, or we cannot just stop the wind, it would be better if their pollinating agents, that is the insect which pollinates the Eichhornia, if their pollinating agents are reduced, their sexual reproduction could be minimized.
Is it anything due to the addition of nitrogen or phosphorus into the water? Contamination
no, I don't think
of the water?
that they grow because of an addition of extra nutrients. There are they grow taking the nutrients from their aqua system or wherever they are infested. They take the nutrients directly from the water body itself. I don't think there requires any additional nutrients to be added for their growth.
Yeah, okay. From my knowledge, usually the water doesn't contain the phosphorus and nitrogen to a large extent. So due to the added pollutants into the water, the concentration or growth of the water hyacinth is more. It gets on to the another level. So if we are using, as you said, if we are using some pesticides or fertilizers to stop its growth, uh, I think if do the pesticides or fertilizers do not contain any nutrients that encourage the growth of water hyacinth or icornia.
I don't think so that the pesticide actually try to degrade or decompose icornia, but if pesticides are used directly, it will also affect the other organisms, including the microorganism that is in that particular water body. And also, if it's a residential area, if you use the pesticide into a residential area, we all know what will happen, right? It will adversely affect the other water bodies near them.
No. Was if the pesticide and fertilizer had phosphorus or nitrogen content in it, that would in addition help to the growth of water hyacinth or icornia. So that would again result in the growth or maximization of the growth. So
No, I was not suggesting for the use of pesticide. Of course, if pesticides are used, as you said, it may contain phosphorus, which may create a further growth in the icornia. But as I said, this I would suggest the use of Gliricidia sapium, which is a leguminous plant. As I said earlier, it is a leguminous plant which has an insect repellent, antimicrobial, antifungal properties. So if we take the insect repellent property, if we use that particular application to be applied in this icornia's growth, it would be favorable for not favorable for the icornia for their growth.
So that will be limiting the agents.
Yes, it would be limiting their pollinating agents.
Yeah. Okay. Thank you so much.
Thank you.
It was nice. Your presentation was really nice.
Thank you, ma'am.
Jessina, I would like to ask you one question. So when you suggesting that Gliricidia is very effective in controlling the pollinating agents, so have you done any laboratory work or have you conducted any practical work as such, or is it only your suggestion?
Yeah, it is my suggestion till now, and of course we all know now we are in the case of pandemic. I couldn't do any laboratory works related to this, but in future I would definitely love to conduct a research further. But till now I have just a suggestion, ma'am.
So you might have had undergone or read through some already reported works regarding the insecticidal properties of this plant?
Yes, ma'am. I have gone through several articles regarding the insecticidal properties of Gliricidia sapium, and I could conclude that it has an excellent insecticidal property, and also Aedes aegypti mosquitoes. As we all know, Aedes aegypti causes several severe diseases, severe infectious diseases, and it is found that these Gliricidia sapium plant was able to control them, and thereby it has certain insecticidal, pesticidal, even rumaticidal properties, ma'am, rodenticidal properties.
Okay, so how do you actually plan to use it as an insecticidal agent?
Yes, ma'am. We have a lot of way for the extraction of Gliricidia sapium. To take the extraction from Gliricidia sapium plants, there are conventional and professional or laboratory technique. The conventional includes how our farmers did it. That is by directly taking the leaves of Gliricidia sapium plant, just by squeezing it, taking its liquid, then diluting it with water, and we can use it like that. And if it is done in a laboratory, there requires some methodology, so some steps for their extraction. That is, if it's prepared in a laboratory, first we need to take the fresh leaves, then air dry it in the room temperature. Then we have to grind it using a mortar and pestle. Then, if saying about the amount, 1 gram of this grounded leaves, the grounded dried leaves is taken, and it is soaked in 5 ml of 80% methanol. Then it is extracted. This crude is extracted by using a Wortmann #1 filter paper, and this filter, to make it more concentrated, this filtered product was
again taken into an oven at 40 degrees Celsius, and finally we will get 63.5 gram of the extracted green crude, ma'am.
Okay, thank you.
Thank you, ma'am.
Well presented. Thank you.
Thank you, ma'am. Thank you so much.
Okay, Jasina, so you haven't evaluated it directly.
No, ma'am.
Since from reading literatures.
Yes, ma'am. I have looked for several literatures which I could take into this one, because since I'm a undergraduate student, we can't do that much experimental things like PG students. So I haven't done it,
you're
but
saying that the leaf extracts from this Glycidia could be effective in eradicating this
yes, ma'am. That by means I said the extraction taken from the leaves has an insecticidal property which could be used to avoid the pollinating agents from pollinating Eichhornia.
So, in your opinion, how should we do this? How will be the application of this Glycidia extract to this existing water surface?
We can take any method, but I think the conventional method is more easy, and everyone can do the conventional method. If they have the plants, everyone can do the conventional methods as the farmers do. So, but for that we have to make sure it is 100% perfect, or 100% it gives success rate. So it must be taken into only through practicals. I can just say that.
But one thing you should notice is that the main mode of reproduction in Eichhornia is not sexual. It's by vegetative means.
Yes, by asexual reproduction through budding.
Through stolons, offsets, etc. That's why just they break it and getting developed into new individuals. That's the reason for its rapid increase.
Yes, ma'am.
It is rapidly increasing and covering all the water bodies. So I don't think it will be effective as it focuses only on the sexual reproduction.
Yes, ma'am. But if the sexual reproduction could be minimized, I can say that their rapid spreading could be minimized to some extent. I am not saying to completely eradicate the Eichhornia's infestation, but we would be able to just minimize the spreading of Eichhornia.
Okay, good.
Thank you, ma'am.
Thank you, ma'am. Thank you, Jasina. I now invite Mahasina, number BOT UG 0011, to present the project work.
The slides are here.
Hello, Mahasina.
Could you please tell whether the slides are visible?
No, slides are not visible.
Visible now, ma'am?
No, it's not.
It was, ma'am?
Yes, yes.
Could you please tell me whether am I audible? No.
No, the sound is breaking.
Messina, hello?
Now is it audible?
No, you are not audible. Your sound is breaking.
Am I audible now?
Am I audible now, ma'am?
Yes, very much.
So, shall I?
Yes, we can start.
Okay, good evening everyone. I am Asina and and I'm here to present the project documentation of University of the Democratic Plan to College Campus, University College for Women Apura, and this gives the overview of the project. Here you can see the introduction, objective, significance, materials, and networks.
Excuse me. I think
okay,
there is a net issue. There is breaking in your voice.
Okay, we are having some net issues over here. Am I audible?
No.
Can you please tell whether it is audible?
Hello, Messina. Actually, your voice is not clear. It is breaking. I think there is some network issue in your area. So, can you just shift your area to some other place where there is enough range? And also, we cannot see your slide. Have you started sharing it?
Yes, ma'am. I have started sharing.
Actually, but your sound is breaking. It's not clearly audible.
No. Start with once. Ma'am, is it audible right now?
Hello, Messina. Are you talking now? Are you speaking now? We cannot hear you.
I think she has left.
She just left the meeting just now, ma'am.
Oh, okay. So, I think there is some network issue.
Okay, we'll continue with
We
our
can
next
proceed with the next presentation, I think.
Okay, I invite Aishwarya K, number BOTUG0012, to present her project work.
Ma'am, is it visible?
Yes.
Good afternoon to one and all. Here I am Aishwarya presenting on the topic isolation and characterization of novel bacterial strains from soil sample and putative species identification through APIS online microbial identification tool. As we know, soil is essential for the maintenance of biodiversity above and below ground. The study of organisms in soil is known as soil microbiology. The identification of bacterial isolate can be performed via characterization of morphology by gram staining and various biochemical tests. The present study was designated to isolate novel bacterial strains from soil sample and their morphological and biochemical characterization. In overall, we had identified 4 bacterial isolates and their biochemical properties and varying morphology were identified. Out of 4 bacterial strains, one of the strain were analyzed for their characteristic pigment production, biofilm formation, and antibiotic susceptibility. The overall aim of such a study is to gain knowledge about more soil-resident microbes. Moreover, it helps to identify strains producing novel bioactive compounds for industrial application or strains with potential to become opportunistic pathogens. And these were the aim and objectives. Isolation of bacterial strains from soil sample by serial dilution and plating method, and then characterization of bacterial isolates based on colony morphology and gram staining, and their biochemical test and enzyme production, and evaluation of antibiotic sensitivity, antagonism, and biofilm production by SS4 strain, and pigment produced by SS4 strain were UV visible spectrophotometry and optimization of conditions for pigment production, and these identification of SS2, SS3 and SS4 strains via APIS online microbial identification tool. And these were the materials and methods. The various biochemical tests various biochemical characterization of isolates were indole utilization test, citrate, oxidase, catalase, urease, TSI, and the analysis of enzyme production tests were starch hydrolyze, gelatin, casein, tween 80. Salt tolerant assay was done. PVK solubilizing phosphate test was done. Antagonism assay, biofilm, antimicrobial test, and pigment extraction using spectrophotometer, and the bacterial identification using APIS online tool. And these were the result of morphological characterization of SS1, SS2, and SS3. SS4 where the color was red, and other 3 was whitish, ivory, milky respectively. These are the result of gram staining of 4 isolates. The various biochemical test result was this: indole, methyl, citrate, urease, oxidase. Starch hydrolyzing, these were the result, and here SS2 were hydrolyzing. The all 4 strain was growth were observed, but SS2 was hydrolyzing. There is the result. And phosphate solubilized PVK medium were used, and the 4 was growth were observed, and SS4 was red color and its pigment was lost. And the next test was biofilm production test by SS4. Formation of biofilm was observed, and these are the antagonism assay using streaking method, SS1 versus SS4. Here intense pigmentation was observed. It was not inhibitory towards SS4. Next was SS2 versus SS4. They were more competitive. It was more competitive, and SS3 versus SS4 was effective or not observed. Inhibitor was not observed. And these are the salt tolerant assay result. 1% NaCl, 5% NaCl, 10% NaCl. The growth was seen, and in 1% NaCl, all the 4 strains were grown. And in 5% NaCl, SS1 and SS2 and SS3 were. SS3 does not grown, and in 10% NaCl, no growth observed was seen. And these are tween 80 hydrolyzed for lipase production. The for 1, 2, 3 SS1, SS2, and SS3, the lipase production was not observed. The growth was not seen, but in SS4, the growth were seen. The lipase production was observed because the precipitation holes were found. Therefore, growth were observed. Lipase production was observed. And this is the antibiotic sensitivity test. Antibiotic use gentamicin, tetracycline, amoxicillin, and ampicillin. Amoxicillin was resistant, other three were not. These were the optimization of parameter for pigment production for SS4. At 35 degrees Celsius, the pigment was lost. It became orange color. So the pigment was lost at high temperature. And through online bacterial identification tool, these strains names were obtained. The SS1 was unable to identify, and score was nil. SS2 was Phenibacillus, SS3 Bacillus, and SS4 were Serratia. These were the screenshot of ABS online tool result of 4 strains, not 4 strains, 3 strains. And I conclude that the study was attempt to screen novel bacterial strain from soil with diverse biotechnological application. The 4 bacterial strains from soil sample were characterized through morphology, gram staining, and biochemical test. Through API's online tool, SS2, SS3, and SS4 as Phenibacillus, Bacillus, and Serratia respectively. The production of biocolor by soil inhibiting bacterial strain SS4 was studied. The red pigment production was found highest at 20 to 30 degrees Celsius. And the online tool served as a first hand for putative identification of microbes. However, strain identification through molecular techniques were warranted. The current result can add up to knowledge on soil biomolecules and the novel biotic compounds produced by them. And these were some of the references. Thank you.
Thank you, Aishwarya. The session is now open for discussion.
Aishwarya, from where you have collected your soil sample?
Ma'am, dumping site of the kitchen waste. Soil from dumping site. Yes, ma'am.
Can you name the online identification tool you use for study?
Ma'am, ABIS online tool. Advanced Bacterial Identification Software.
Thank you.
Aishwarya, how long it took to complete your work?
Ma'am, 2 months, but the result was not obtaining. Therefore, I tried this soft online tool for naming the species.
Okay.
Contamination was occurring. That's why 2 to 3 months, ma'am. 2 to 3 months.
Okay. So, from you are collected from the place where kitchen waste are dumped.
Yes, ma'am.
So, what is the purpose for collecting samples from there?
Is there any useful soil? Is there any bacterial isolate that are useful for industrial application? To check for that.
By industrial application, what do you mean?
Ma'am, that my SS4 strain, that is Serratia, which has a biocolor. So it's an industrial application for biocolor production, pigment production bacteria was. So it is applicable in industrial field. To identify, is there any?
Is that the only application you find?
Yes. My PG, in my UG project, I have done that.
You have tested various enzymes, enzyme production ability also. What about that?
Okay.
Enzymes are commercially very valuable.
Yes, ma'am.
Okay.
Phosphate soluble. There are 4 strains of phosphate solubilizing bacteria.
What? Some more louder, please.
PVK I have used PVK medium phosphate solubilizing materials. The 4 strain where they were solubilizing in phosphate and lipase, and they are also lipase production for SS4 strain.
Okay, what was the advantage of getting phosphate solubilizing strains? No. Okay, in your work, I'm saying that you have done a lot, but you have to focus on one or two things, and you have to work deeply on that those factors. You understood what I'm saying?
Yes, ma'am.
That's what I have to advise you.
Ma'am, it was a group project.
Okay, that's not a problem. But I'm asking you, what was the major objective when you started this work? We should have a focus, a sharp focus for something. We should do something for that is useful for our society. Okay, so our objective should be sharp.
Okay, ma'am.
Okay.
Good evening all, myself Manju Thomas. My presentation topic is Tree Diversity and Carbon Sequestration Potential, a study in Alfonso College campus. Climate change and harmful role of greenhouse gases in the changing temperature is a burning issue today. So the present project in Alfonso College campus is an attempt to identify the tree diversity along with a study on their benefits and to analyze their efficiency in sequestering carbon. Trees are of invaluable importance to our environment and to human well-being as well. So the knowledge about trees in a community will help the inhabitants to promote its diversity and sustainable management. Trees also play an important role in capturing carbon dioxide from the atmosphere by fixing carbon during photosynthesis and accumulating extra carbon as biomass. These are the objectives of our project. The first one, to identify, document and evaluate the diversity of trees in Alfonso College campus, to analyze economic, environmental and aesthetic benefits of trees in the campus, and the third one, to calculate the carbon storage and sequestration by selected tree species. So moving on to the methodology. First is the methodology to identify the tree diversity. Field survey was conducted and trees were identified using pertinent flora. Frequent visits were done to identify the flowering and fruiting seasons along with taking their photographs. Next is the methodology to calculate carbon sequestration. The AGB, above ground biomass, was calculated using the allometric equations developed by Uday Kumar et al. For the tropical dry forest. These are the equations used. The first one, AGB, above ground biomass, was calculated using the allometric equations. That it is the above ground biomass. Here, 2.2014 and 1.0615 are constants. The next one is BGB, it is below ground biomass. Then TB, it is a sum of AGB, above ground biomass, and BGB, below ground biomass. Next is carbon storage. Carbon storage in the tree was estimated by multiplying the biomass of the tree by 50%. Because for any plant species, 50% of its biomass is considered as carbon. The next is carbon dioxide sequestered. Carbon dioxide sequestered in the tree was determined by multiplying the weight of carbon by 3.67. That is weight of carbon in the tree by 3.67. Here 3.67 is the ratio between carbon dioxide and carbon. Next is the results. First we'll show the results of the diversity of trees in the campus. We identified almost 315 trees belonging to 22 families, 44 genus and 47 species. The richest families identified were Fabaceae, Euphorbiaceae, Moraceae, etc. And the abundant species among these were Tectona grandis with a number of 107 and Sweetenia mahogany with a number of 88. Now take a look at the table. Here the highlighted ones are the most abundant species. Yeah, so the table goes down in a descending manner. Yes.
So in this table, it is clear that the family Fabaceae is the richest family. Here, the 40th one, Peltophorum pterocarpum, belongs to Fabaceae. 43rd one, Pterocarpus marsupium, belongs to Fabaceae, and 44th one, Saraka asoka, belongs to Fabaceae. Tamarindus indica belongs to Fabaceae likewise. And these are the photographs taken by ourselves from our own campus using our own mobile phones. Yeah, this is plate 2, and this is plate 3. The first one is Mangifera indica, shown in this figure, in this photo. Plate 4. Next, plate 5. And this is the final plate 6. So next we are moving on to the second objective. The result of the second objective, the benefits of trees. Ecological importance of trees. Yes, beating air pollution, beating noise pollution, etc. The tree species like Tamarindus indica, Mangifera indica, etc. are effective in controlling air pollution, which are identified in our campus. Next, economic importance of the trees. Tectona grandis provides the best angiospermic timber in the world. Sweetenia mahogany is also economically very valuable. These trees are also identified in our campus. Next is aesthetic benefits. Trees like Delonix regia and Tectona grandis act as giant sun blockers, providing shade, relaxation, and visual treat in our campus. So next is the results of carbon sequestration value. Before that, remember that carbon sequestration is the mechanism for the removal of carbon from atmosphere by storing it in the biosphere. When trees capture carbon dioxide by fixing carbon during photosynthesis and accumulating extra carbon as biomass, it is called as terrestrial sequestration. Yeah, so take a look at the results of the carbon sequestration value of each tree. Yeah, these were calculated using the equations mentioned earlier. Here, when we see, we can identify a positive relation between the DBH and carbon dioxide sequestered. In the first case, Albizia summon, it has a DBH of 275 centimeter, and corresponding sequestered value is higher, 52,372.11. And Delonix regia also, it has the same value. See, while DBH is decreasing, the value of carbon dioxide sequestered is also decreasing. It is clearly visible in these tables. And coming down, here Nephalium lepacium has the lowest amount of lowest DBH and lowest amount of carbon dioxide sequestered. So summing up the results of the values got earlier, Albizia summon and Delonix regia are found to have the highest carbon sequestration value. Yeah, and Nephalium lepacium is found to have the lowest carbon sequestration value. The trees with higher biomass have more sequestration potential. Woody plants store more carbon than others. And as I said earlier, there was a significant positive relation between the tree DBH and carbon dioxide sequestration. This graph shows you the results of carbon sequestration value of different trees. Delonix regia and Albizia summon has the highest value, which is shown to the bottom of the graph, and on the top of the graph, Nephalium lepacium, it has lowest value. So, we select trees for to calculate the sequestration value of trees, we should select the trees with such specialities. It should have high specific density, fast growth, huge canopy, better climate adaptability, richer litter productivity, and so on. So to conclude this project is very much relevant, as the results of the study may facilitate further planning and decision making regarding plantation in the campus, because there is a need for better management and conservation of biodiversity in that area. Think for it wisely. One tree can make a difference. So when will you plant yours? And thank you.
Okay, thank you, Manju. The session is now open for discussion.
Okay, Manju, nice presentation.
Thank you, madam.
One question. What may be the reason for predominance of Fabaceae in your campus? From your result, you told the Fabaceae members are most common.
Yes, ma'am. Maybe because of the good environmental conditions it has, because because it has no competitors around those plants, and the members belong to Fabaceae could be easily growing in that soil. Yes, ma'am.
Okay. Have you read any literature like that?
No, I haven't read, ma'am.
Okay.
Manju?
Yes, ma'am.
Have you heard the earlier presentation, a similar one to yours?
Yes, ma'am. Yes, ma'am.
Okay. In that study also says that Fabaceae has more impact.
Yeah.
So it is repeatedly saying that Fabaceae is more potential for carbon sequestration. So there may be something. So you have to read more about that in literature.
Okay, ma'am.
Okay. Then what was your conclusion?
My conclusion was that Dendronix region Albizia salmon, which has higher DBH value, has more carbon sequestering potential, and this data will be passed to the college authorities, and they should take necessary actions further while planting new trees.
Okay. Do you find any difference or similarity with the earlier presented work?
Yes, ma'am. In that presentation, she took height as a point to calculate carbon sequestration.
Okay.
And in our project, we didn't take height because our land was not flat. It includes curves and slopes, and using clinometer, we couldn't find out the height.
Okay.
And so that we use allometric equations provided by Uday Kumar et al. In that, DBH was calculated instead of height.
Okay. Next to Fabaceae, which was the next family showing higher impact? Euphorbiaceae. Okay. Do you have more Euphorbiaceae species in your campus?
Yes, ma'am. Makaranga palpata. It belongs to Euphorbiaceae.
Okay, good.
Thank you, ma'am.
Thank you, ma'am. Thank you, Manju. Now I invite Arya Sajeev, number BOTUG 0014, to present her project work.
I'm Arya Sajeev. Today I'm going to present about frugivory and seed dispersal of trees in CMS College area. Frugivores are organisms that depend on fruits for their diet. This is the most basic thing about frugivores, and they depend on raw succulent fruit like vegetables, roots, shoots, etc. And they show high dependency on abundance and the nutritional composition of fruits. They either benefit or hinder the plant, like they either disperse the seed or destroy them. It's like a mutualism. Frugivores consume seeds and disperse by a gut passage mechanism after chewing the pulp of the fruit. Seed dispersal help in escaping predation, competition, and fungal attack. The aim of my study was to study more about frugivores and seed dispersal of common trees found in campus of CMS College. Materials and methods. The study area is CMS College area, and the materials I used were binoculars, camera, and field book and a thread. Okay, the methods I followed were the study period was from December to February. The period was about 10 hours, and the day was divided into 4 intervals, that is 8 to 10, 10 to 1, and 1 to 3, and 3 to 6. For data collection,
and the number of bird species and the feeding visit were recorded on during these periods, and the birds which are pecking and swallowing only were recorded, of fruits were only were recorded. Simply preaching into the tree were not recorded, and this was identified by direct observation and also with the available literature that I used was Common Birds of Kerala by Institute. Okay, the results I got was fruits being fleshy and small and attractive makes the fruits accessible to the frugivores, and availability of the fruiting trees in the campus is the major fruit producing trees are the ficus species. We have Ficus asperata and Ficus bengalensis. These aid as the major food biomass producers of the campus, and from the observation I found that the birds preferred maximum visit during the morning hours than the midday or the evening hours. Okay, among 15 species of frugivores observed, bats, squirrels, Asian coils, and Myna, Rufus trippi made a fruiting visit in groups, whereas others visited individually or with other species. Among animal frugivores, bats and squirrels consumed maximum number of fruits, whereas of avian frugivores, Rufus trippi and Asian coil appeared as the major agents of seed dispersal, and also the most consumed. They consumed most fruits. Okay, total 174 visitors of frugivores were recorded, and in which squirrels are the most represented visitors, and in avian frugivores it was eudaemomus, that is Asian coil, and Dendrocita vagabunda is the Rufus trippi, and Petrobus giganteus is bat, Indian flying fox. It was noted that the individual visited the ficus consumed the whole fruit, whereas that visited papaya only consumed the pulp. From the droppings of avian species, we understood that they aid in the seed dispersal. Also from the scats of Asian palm civet, we understood palm seeds were found. So we understood that they also aid in the seed dispersal. Other interesting factors I observed was the one after other fruiting pattern of the ficus. This is a list of the number of visits made by the frugivores. The Scuridae as the most represented visitors. Okay. This is coppersmith barbet on Ficus bengalensis. They are highly dependent on ficus, and this is Asian coil. They are also dependent. This is Indian flying fox on Ficus bengalensis itself, and this is Asian coil female on Ficus asperata. This is yellow fruits. They produce yellow fruits. This is yellow oriole on Terminalia catapa. That's Betham tree, and this is Rufus trippi. Dendrocita vagabunda in Syzygium. This is rose apple. This is white-cheeked barbet on Carica papaya, and this is red-banded bulbul in Albus samna. This is rain tree, and this is jungle myna eating Carica papaya, and this is a group of Rufus trippi eating Carica papaya. Myna, same jungle myna eating Carica papaya. This white-cheeked barbet. So for analyzing the characters of the fruit, Ficus bengalensis fruits were taken, TS of the fruit were taken, and the diameter was checked. It was about 2.1 diameter, 2.1 centimeter, and the seeds are isolated from the droppings of the avian frugivores, and this is the papaya seeds dispersed by the Indian flying fox, and these are the partially eaten food by Scuridae squirrels, and these are the scats of Asian palm civet. If you clearly notice, you can see the palm seeds inside it. And for further discussion, as I said, the green campus of CMS College is really rich in fruit-bearing trees, and this attracts major frugivores to this area, and a wide variety of fruit-bearing trees are there, like Ficus bengalensis, Ficus asperata, Carota urensis, Albus samna, Terminalia catapa, etc. As I said, among animal frugivores, bats and squirrel consumed maximum number, and in avian frugivores, Rufus trippi and Asian coil consumed the maximum. Mammals are unlike birds; they have hands, teeth. So they are the major frugivores than the avian ones. As ficus species are major producers of fruit biomass in the study area, they are an important fleshy fruit resources for the frugivores. Also, coppersmith barbet showed a high dependency on the figs, and they appear to be the fig trees appear to be the keystone species in the campus for some species. So for the conclusion, in my observation, I found that birds preferred more visit during the morning hours than the midday hours. Among 15 species of frugivores observed, bat squirrels, Asian coil myna, and Rufus rapi made their visited in the groups, and others individually or with other species. It was observed that among animal frugivores, bats and squirrels consumed more number of fruits, and Asian coil and Rufus rapi consumed the more number of fruits among avian frugivores and appear to be the major agents of seed dispersal. From the present study, it was understood that frugivores are attracted by the fruit color, fleshiness of the fruit, and bright, as I said, bright color, size, etc. The study also revealed that ficus is a major keystone source of biomass in the campus and appear to be the most important food resources for both avian and mammalian frugivores, and may be a keystone resources for some species. The loss or decline of ficus trees could lead to the decline in the population of this species. Okay, these are some references. Thank you.
Okay, thank you, Arya. The session is now open for discussions.
Arya, what is the relevance of your study?
Ma'am, relevance of my study is about the fig trees. If birds like megalme, like coppersmith barbet, it shows high dependency on fig trees. So conservation of fig trees is really important to serve the diet of the frugivores. So I think that is more relevant, and you can also study more into the amount of things they amount of fruit biomass they ingest during the period of the whole long day. So I think it is really interesting to study more into this. So that's relevant.
Okay.
How long it took to complete your work?
Ma'am, I started in December and I ended up like mid of the February.
Okay. So, how many months?
Sorry, December, January, February, 3 months.
Okay, you are directly observed.
Yes, ma'am, I was directly. That was a very much tricky thing to my patients. It taught me a lot of patience in direct observing the birds.
Okay.
It was actually fun during the period.
It shows your interest on this topic.
Yes, ma'am. I was not too much into this topic. Actually, I was astonished when I got this topic from KFRI from Srikumar sir. So when he told this topic, I was actually said I don't know anything about this. So that's why you are given this topic. You should study this. And my HOD was more showed much interested in bird watching and all. He is a professional bird watcher, so I had a great help from him. So now I started this as my hobby. Now I watch birds while I travel and while I move on like that. So that's a great thing.
Okay.
Now I got a new hobby.
So you're concluding that fig plants have a major role?
Yes, ma'am. Fig plants act as a keystone species inside our campus. If you know that we have more fig trees.
By saying
keystone, it means that one organism is dependent on the other. Like if a keystone species is removed from an ecosystem, overall change would be happening in an ecosystem. Like it would depend the life of the other. Like maybe decline in the species.
I'm asking what all species are depending on this fig tree?
Yes, ma'am. I would say Asian coils are mostly dependent, and megalme, coppersmith barbet, white-cheeked barbet, and Rufus rapi, and I don't know if you know alkali or the squirrels and others. Most of the as our campus have like 3 or 4, I guess 4. I guess 4 trees are there uh, of figs, different species like Ficus bengalensis and Ficus asperatus is also there. So
Okay.
mainly I have seen squirrels and bats as highly dependent as in mammalians, and others like Rufus revi and Asian coils are always seen in the tree.
Do you know one thing? Squirrels have a major role in this tree plantations. In our in your CMS campus also, there are so many trees that have come over because of these squirrels.
Yes, ma'am. We have monkeys also in our campus. If you have seen, Elizabeth ma'am have seen, right? Monkeys also in our campus. Our campus is attached to a virgin forest, so we have monkeys too. Monkeys, squirrels, and Asian palm civet too in our campus as mammals, and they all aid in this seed dispersal, ma'am. However, yeah.
Okay.
But I didn't find any monkey eating this pig and all. I heard my sir telling me that they depend on palms. Monkeys depends on palms. Yeah.
Okay. Okay, good.
Thank you, ma'am. Thank you.
Thank you, ma'am. Thank you, Arya. So with this, we come to the end of the presentations. Thank you all. Now I invite Dr. R. Nisha Nair to propose a vote of thanks.
Good evening, everyone. Respected resource person of the day, Dr. Elizabeth Cherian, Research Committee member from the Department of Botany, Dr. Diana KG, teachers and delegates from different parts of India, and my dear students. It's my pleasure to propose vote of thanks to all those who made this program so fruitful. First of all, I extend my deep sense of gratitude to Dr. Elizabeth Cherian, Assistant Professor, CMS College, Kottayam, who was so generous enough to spare her valuable time with us today. She not only evaluated the presentations critically, but also gave her valuable suggestions, which I am sure would help the students improve their research works in future. Thank you, ma'am. Now I thank our principal, Dr. Rita Latadi Kuto, and Research Committee Coordinator, Dr. Bhagyadi, for taking up such a wonderful initiative and for all the support and cooperation extended to us for carrying out this program. I thank all the student presenters who are actually the essence of this program for making this event successful. We hope that this platform would surely have helped everyone in acquiring new scientific ideas. Thank you all. I thank the teachers and students from other institutions for their participation. And finally, I thank all the teachers and students of St. Joseph's College for Women, Alappuzha, for all the support and cooperation. Thanking you all once again. Thank you.